You.
Hello, everyone, and welcome back. It's my pleasure to introduce our next keynote speaker, the fourth keynote. We have Jeremy Schmutz from the HudsonAlpha Institute of Biotechnology. He's a faculty investigator there, and really has been in this capacity has been using PacBio for a very large range of organisms, which I think he'll all cover in his talk. So it's going to be a very wide breadth. He's been a very early adopter and champion of the technology. So I think you're in for another real treat. And with that, I'll give it over to Jeremy.
Thanks, Jonas. I appreciate it. PacBio gave me an opportunity to talk about the different kinds of things that we're doing here. I'm going to go ahead and share my slides here, and we'll get going. Let's hope so. That was super smooth. Okay, thanks again, I appreciate it. I'm going to talk about both our work in humans and our work in plants and other organisms today. I'm going to start with humans. These are my humans. They don't quite look like this anymore, and it's, I think, pretty questionable whether they actually share 50% of my DNA or not, but that's a different story. I actually got started working on the Human Genome Project along with lots of other old fogies or old dinosaurs, as Evan and I like to call them in the genome world. And this is me about 20 years ago on something called a 3730, which seems like a foreign box these days. It actually used capillaries to sequence things. And this is our group at the Stanford Human Genome Center. We worked with our partners at the Joint Genome Institute, which is a Department of Energy funded facility, to sequence 3 chromosomes, 5, 16, and 19. 5 is my favorite, not really for any specific reason, but it's the biggest one. What made it great, the human genome, the original human genome? Well, one of the things that made it great was single haplotype. And so we actually got everything into little packets called clones, and then we sequenced the clones, and so they were individual single haplotype, and we put them back together again, just like a big puzzle, into the human genome. And so that's really helpful because it got rid of any of the confusion of sequencing 2 copies of DNA at the same time, which is what we always struggle with when we're working on whole genome shotgun sequencing projects. So the other thing that made it great was this thing called manual inspection and finishing, quote unquote finishing, where we had people who actually went through, people, including myself, went through and did computational finishing and looked at almost every base pair in the genome and ordered new sequencing reads and went through and did reactions, then brought those back in again and did it again and did it again and did it again. This is a view from David Gordon's concept, which we used almost 20 years ago. You can still use it today. You can even use it with some tips and tricks with HiFi data if you want to look at variants, and I encourage you to talk to David about that. And so we go through and we finish these clones and we'd improve them and we figure out where all the problems are and fix them. There's a great part right here where I type in the end cut site here. It's a Hindi 3 cut site. And we did that on thousands of clones over the Human Genome Project also. So this is what it was like really to sequence a genome. And some people didn't get to experience that. Now you've had like almost a whole minute of it. And so I typed in the dang Hindi 3 so many times that we just put it on our license plate because it was easier to remember there. And it's like muscle memory now. And finishing was really like an art. You would go from a clone that looked like this. This is showing paired ends across the circular clone and all these gaps in there. And we do lots of sequencing reactions and lots of manual labor and clean it all up and make it really look really nice and get it high quality. Same thing for repeat. We'd go in and we'd sort these things, like sometimes for hours or even days, you'd spend sorting a repetitive region of a clone like this with all these so it looks really nice here. There's a 20 kb repeat in a clone and sort it all out. So that was a lot of work. It was a lot. So some stuff happened since then, 2003 to 2017, right? We went to the 454 days from the Sanger days, and then we got Illumina, which gave us huge amounts of data, but really short reads. And then the RS2, which is where PacBio really started to become useful for genomes, and you could get longer reads, and then onto the PacBio SQL platform, where we started to be able to actually really produce a lot of genomic data from there. And so costs there are human genomes, right? $30 million was about what it cost at the end of the Human Genome Project to resequence a human and generate an assembly, down to about $100,000 on the Sequel 2. At the same time, our group worked a lot on plant genomes and environmental sequencing as part of the Department of Energy Joint Genome Institute. And then also we moved our group from California down to Alabama, down to the HudsonAlpha Institute for Biotechnology. And down here in Alabama, we've got a lot of space compared to California. So we're able to put this 3/4 mile long double helix in place there. And you can actually go for a good exercise at lunch. You can walk the Watson and the Crick Strand and really get a good experience there. But the HudsonAlpha Institute was a nonprofit really with these 4 areas: genomics research, clinical genomics, education outreach. We do a lot of education outreach for Alabama, and then economic development. We are co-located with almost 600 employees and 40 associate companies working in biotech, a lot of it related to genomics. And our goal is to apply genomics to make the world a better place. Actually, that's not our goal. It's a lot longer and less interesting than that, but that's really what we started the Institute for and continue to work on. So HudsonAlpha's got a rich history applying genomics to human medicine. We're part of the ENCODE projects and chromatin mapping projects, done a lot of work in cancer genomics, understanding cancer biology with genomics, de novo disease identification, identifying new variants that cause disease, lots of whole genome association studies for complex traits. And we also have a genomic medicine clinic here where you can go if you have a genomic disease or a putative potential genomic disease and work with Dr. David Bick to try to identify the underlying cause of that disease. So we've worked on plants, I just said, and other things for a long time. And so one day we were kind of sitting around faculty thinking, what could we really do to try to bring this new technology that we were working on in plants to humans? And so this was the kind of question: what if every patient had a genome? And so no longer were we talking about, panels and targeted assays and exome capture and whole genome shotgun. What if we just produce genomes for people? This is a project collaboration between Greg Cooper's lab and Jane Grimwood and myself, mostly led by Susan Hyatt and Lori Handley, and lots of other collaborations from our folks and collaborations on the PacBio side. So we built this on top of the CSER project, which is a project funded by NIH, led by Greg Cooper. It's now looked at 1,100 affected children with developmental delay diagnosis using whole genome shotgun sequencing. And these are the results and findings. About 58% of them received no diagnostic result after WGS with Illumina sequencing. And so we were hoping to try to do some better with PacBio. So we did the first thing, which was build a genome, because I hadn't really looked at a human genome in a long time, and we wanted to sequence one. So we took one with about 80x of long reads on 32 SQL chips. That tells you when it was. Assembled it with MeCat, Illumina error corrected and integrated. We got about 3 gigs of the genome out, 3,000 contigs. That was pretty good. I thought it was really nice looking. 11 meg contig N50. And it looked kind of like this. This is just a dot plot, which shows you, if you're looking at a dot plot, you want to look down the diagonal here, shows you how closely related 2 pieces of sequence are. In this case, this is a build on chromosome 20 versus HD38, and it looks pretty similar. Not too bad, not a lot of changes and differences in there. So I've got some take-home sequencing facts, genome sequencing facts here, and this is number 1. This is a pretty obvious one to start with. Longer sequencing reads actually equal better assemblies. You don't want to see what an Illumina human genome assembly looks like, and you certainly don't want to use it in a clinical setting. So about the same time as we've been working on that project, Sequel 2 emerged on the scene. This is our first Sequel 2 here, and this is the first Sequel 2 customer run there, where we produced a pretty exciting sorghum genome, I thought, and then started doing some clinical research-based sequencing there with HiFi. We worked through early access and then through the Sequel 2 collaboration program over the first year, and are about something like 15 to 25 gigabases a cell. Of course, it depends on what you're running, what the quality of DNA is going into your libraries. So this is what the basis is for that. HiFi, what is it good for? Well, most people know that it's not actually absolutely nothing. It's really good for things like allele detection for nucleotide variants, but also for indels and insertion deletions. I think it was noted yesterday that it's really good in the mid-level insertion deletions. This is uncombable hair syndrome, TCHH, and this is 2 separate alleles that are different than the reference sequence and deletions here. This here are the Illumina reads, just so you can see what Illumina reads look like in this region when you get into this 18 nucleotide repeat region. And so take home number 2, longer accurate reads are better for SV calling in difficult regions. Hopefully I'm not telling you anything you haven't heard before. So we went ahead and sequenced 6 separate probands from CSER, plus their parents with HiFi at 12 to 24x. We called SVs with PBSV and sniffle survivors pipelines. That's just an example of a child-specific de novo mutation here, a deletion. And then we called them across all of our probands and looked at that, and you can see that difference here is that PBSV calls about 5 times as many variants. That's because it's calling to a very much shorter level of indels than sniffles is, but you can see that they call quite a few across there. When we go back and we say, well, how many of these are truly de novo? About 62 are de novo from each one of the calling methodologies. That means 62 times we've got a structural variant that's callable with PBSV or sniffles that occurs in an individual offspring. They're not exactly the same 62, but there's a significant overlap between the two of those. Some of these results are, and what I'll talk about following, are summarized in a bioRxiv paper led by Susan Hyatt. So on proband 6, on the X chromosome, we identified a de novo insertion. This insertion is in the CDKL5 gene, and what it is about a 7 kb insertion that's hopped in the middle. But it's also carried along with it, like transposons do, a copy of the exon 3 CDKL5 exon 3. So it occurs in 2 locations there, adding to the protein and reducing its effectiveness of transcription and potentially production. This is also the gene that's been called out as causing Rett syndrome. We don't have a complete Rett syndrome pathology for this proband patient, but we have very similar symptoms on there, and so we're confident saying this is a likely pathogenic insertion. What's interesting about this one is it's a mosaic, and it occurs in about half the cells in the paternal line, and so it's effectively invisible to any kind of filtering that you might do on Illumina structural variant finding for this. So but when HiFi gets powerful is with the new assembly methodologies. Most of these things have existed for less than a year. Some of these have really existed effectively for 6 months at this point, working through like HiCanoe and HiFi ASM, where they're really starting to become powerful working with this data, including things like introducing trio binning approaches. And so that's the approaches that we've taken to try to understand what's going on in these probands that we've resequenced with HiFi. So you can see the difference here. Long read assemblies, we generate about 3 gigabases of data, 12 megabase contig and 50. HiFi, we generate about 6 gigs of data, and that's with the trio binning approach, splitting it into individual haplotypes. So with HiFi, you can get access to single haplotype data, and it's super important for medical things. From the human side, it's super important in understanding how biology and life works and how the genetics interacts with their environment across all organisms. So these are what our de novo builds look like. Got about 3 gigs out each one of them here, 342 contigs to 1100 contigs for each individual human genome, 18 to 49 megabase contig N50s. The best base pair accuracy was our highest coverage. That was still 1 indel error in 86,000 bases, or 34,000 errors in the genome. For human, we finished to something like 1 in 100,000, although the nominal bottom rate was about 1 in 10,000 genes. And so I encourage you, more coverage does equal higher accuracy. More coverage also equals higher continuity. So when you're trying to generate a genome, the more coverage you have per haplotype, the better. So more coverage, I just said it already. More coverage per haplotype equals higher continuity and a better assembly. So are these assemblies any good? Here's chromosome 5. Again, I mentioned my favorite chromosome. This is a proband, chromosome 5 versus B38, and you can see the solid, amazing comparison. It's nearly identical all the way down the board to B38. You can't see any real structural variations on here at all. You can see that on the PacBio, we're recovering some version of paracentromere and centromeric regions here in human versus our B38, where we were bound by our clone-based sequencing and ended up entering into, but not getting into the middle of any of that stuff. You can also see here, this is the SMA region that's been mentioned a few times, one of the most difficult regions in the human genome because it's very large tandem repeat that spans multiple megabases, but it's got some really interesting pieces in there that are over 100 kb with 1 to 2 nucleotide variants in there. And this is kind of what it looks like on our HiFi assemblies here at about 20x haplotype. And you can see it's not perfect in any sense. We are definitely generating a variance of SMA, but we're not getting through this region with our current versions of HiFi sequencing. So one of the things that we identified using this methodology of building these genomes out is a complex de novo mutation in proband 4. This is one we ended up taking a higher coverage because we couldn't figure out what the heck was going on really on the patient sample. And what it ends up being is 22 megabases of chromosome 7 has moved to chromosome 9, and 3.6 megabases in translocations back on chromosome 7 with an inverted repeat on the front part of that. And so this was a pretty interesting finding. It turns out that this patient had likely not had a lot of cytogenetics work done, and on an array based assay, this would appear to be neutral because we have all the DNA that's present there. However, on further observation, we also found some interesting issues with chromosome 6 here with a large paracentromere conversion, and then also a fractionation that might be similar to something like a chromothripsis activity, all derived by the parental line, where several regions are affected in this proband. So we've got multiple genes affected here that could cause the pathology. One of them on chromosome 9 has been associated with neurodevelopmental delay disorders, and we can verify that there's issues with gene transcript production in that one. However, there's also chromosome 6 genes that are potentially affected, and several genes, in this case 6 on chromosome 6, that are affected by this. So there's multiple genes that could lead to the pathology for this proband. So we ended up with, out of this set of 6, 2 out of the 6 WGS negative patients now have potential diagnoses or follow-up from HiFi sequencing. So what's the cost of this? I think you heard $3,000 this morning. So I'm going to say $6,000 to do SV calling here with HiFi, 28x HiFi, about $13,000 to show for these mid-tier assembly kinds of things that I was just showing, about $30,000 for the El Primo reference with some other data to really be able to build out an accurate, mostly haplotype resolved human genome. Obviously, specific annotation and variant validation costs more. So my take-home number 5 is it's possible to largely replace our nearly $5 billion effort with $30,000 today, thanks to PacBio HiFi. I'm not really sure whether I should be happy or sad about this. Technology keeps marching on, so it's pretty exciting. But human genomes are really relatively simple. They got 2 chromosomes. They're super boring with like 1 in 1,000 variants between those. And look at this. This is a dot plot, and you can see the background repeats here in humans on this piece of chromosome 5. This is the top half of chromosome 5. And you can see, other than the SMA region showing up here, there isn't really a lot of repetitive content in humans. So plants are more complex. We can start with those chrome 5A and 5B. But our haplotype variation can be as high as 1 in 33 bases or even higher, depending on the plant you're working from. We can also have a 5C and 5D to make a tetraploid, or 5E and 5F to make a hexaploid, or even more, decaploids, dodecaploids, crazy numbers of copies of chromosomes. And the repeat content is way higher in plants because they're continually evolving. Humans are bottleneck species, you know? I mean, we're not really evolving anymore. That's pretty clear. But plants continue to evolve and produce offspring. And all this black stuff here is all repetitive content in the paracentromere, subparacentromere. There's lots of genes that are being formed and lost in these regions. A lot of the plant work that we do here at HudsonAlpha is part of the Joint Genome Institute, which is a Department of Energy user facility for genomics, where people can write and applications and then work with us on plant genomics. A lot of our mission science is around cellulosic biofuel feedstocks, carbon cycling, and DOE emission science. So we collaborate with lots of groups around the world, and I wanted to mention that some of the data I'll be talking about here comes from our production groups in Berkeley, run by Yuko and Chris. And then this is just some pictures of our flagship genomes. These are the ones that we've worked on now from Sanger days all the way through now to PacBio HiFi days, and we've put a lot of effort into them over time. We've sequenced them a lot, so we know a lot about them. So what has PacBio enabled in plant genomics? Just to give you a little bit of the work that we're doing right now. This is Switchgrass, and Switchgrass is a perennial plant that once was the home, once lived where I came from in Illinois on the Great Prairie. And this is a project working with Tom Younger at UT Austin, and lots of people in our groups, including Jerry and John and Sujin, who do a lot of analysis work, and Jane's group, who produces a lot of the data for all these projects, and then Shu, those are our LB&L group in California, works on plant annotations. And it involves lots of partner labs. I'm just going to give you a little tiny sliver of the genomics parts that we're really responsible for as part of this project, but it's a big project with lots of effort across the US. So what about Switchgrass? Switchgrass, I said, is native to North America. It has a widespread distribution here across the entire eastern half of the United States. It's a C4 perennial. It's highly productive, but it's unfortunately a tetraploid and an octoploid and an otherploid, like lots of these highly productive plants are. And it's outcrossing wind pollinated, which means it's hard to do genetics with. It's been grown for a while for agronomy work, and it is pretty productive in marginal lands, and we view it as a potential biofuel feedstock. And you can see the difference. This is switchgrass that grows in the north of these short buggers here. This is switchgrass that goes in the south here, and it produces a lot of biomass over time, but doesn't have a lot of cold tolerance in the south. And so we're really interested in kind of transposing the traits and getting the cold tolerance of the north into with the biomass of the south. And over time, switchgrass, it's a native perennial wild plant we're working with here. It's been compressed and expanded over time with the with glaciation, and then pushed into glacial refuges. So there's a lot of mixing that's gone on in the genomes over the period. So this is our Switchgrass V5 outbred tetraploid PacBio. This is this is a CLR version here, just 146 gigs of data. Over collect on CLR. If you're ever collecting CLR, again, please don't do that. But if you do, 60 to 70 exahaplotype is where you want to be at. So we used a lot of just our existing resources. Just to show you the difference, in V4, we're at 25 kb. This is everything we've done to date for 10 years of work on this. This is our initial version of our PacBio at 4.3 megabase contiguity. And what that looks like is this huge contigs versus our huge number of contigs. And just in the ordering perspective, look at this ordering. This is compared to a diploid Soteria viridis. You can see how great our ordering is showing up in Switchgrass version 5 versus Switchgrass version 3. What were we thinking? I don't know. So we've done a lot of work with switchgrass population surveys. We collected plants across the entire United States where the plants grow, and you can see some dots here showing you those. We've planted these out in common gardens in 14 different locations. We've gone through and we've resequenced all these things to about 50x coverage, a huge data set for a plant project, especially a public plant project. And then this is what some of those look like just for fun. This is a plant that produces an enormous number of seeds. This is a plant that has really thick leaves and waxy coatings on them. This is one of those short buggers that kind of looks like a dachshund with a couple panicles coming out the top. And here's kind of our archetypal switchgrass that we like. It's upright, looks really nice and clean, produces a lot of biomass, and doesn't take up a lot of space. And so that's kind of variation that we're dealing with in these populations. We were able to use our common garden data to assign out different ecotypes. So it's, lowland here, or upland ecotypes are short, and the coastal lowlands are kind of blends, and then the Texas lowlands look like this with the huge ones here. And you can see there's 3 across the United States. This is important for the next part, where I tell you about the genetics, where we split these up into 3 genetic categories, Gulf, Atlantic, and Midwest. And then from those, we're able to use the ecotype assignments to put these things where they go and say something about their phenotype, what the plants look like. And one of the interesting parts about this is that there's a whole section here that we didn't know existed. I mean, we knew the plants existed because people had seen them before, but on the genetic level, we'd never seen this data before, where the uplands, it's an upland ecotype, that's the short kind of stubby ones, but lives up here in the north, but it turns out that genetically it's lowland. And this is coming from that glaciation and collapsing and expansion, and we're intersecting that data here. And so in 2018, we had a really cold winter in Chicago, and so much so the economists felt the need to explain why Chicago is so cold. I don't need the economists to explain that because I grew up outside of Chicago, and I can still feel the cold in my hands with gloves on as I break the ice off my car to try to get the car door open so I can get to work. And it was really cold. Anyway, we had a lot of plants that died, but you can see the plants that survived. We had a significant number of these plants in the north that came from the north that survived, but are really lowland genetic material. And so you can do this kind of thing now with all this resequencing data and a high quality reference genome and do integration analysis. We're actually looking at this purple Midwestern material and the Atlantic population. And you can see some of it showing up in here. That's evolution, crossing and gene mixing, and then adaptive selection occurring as these plants move further north. And it happens over and over again, and it's reduced us to a small amount of Midwestern material in the Atlantic material that has survived. And we can map that back to the genome and identify these regions of the genome that are associated with this climate adaptation and fitness and production applications there. And you can see it's a small amount of the genome that are associated with any of these kinds of exciting things. And it's occurred over the last 8,000 generations. If you put this back on a plot, you can see here that the further north you get, the further more Midwestern material you get as intergressions in the East Coast material. And for us, we've got these really exciting plants here that are genetically equivalent to lowland or to coastal ecotype, but they survive in the north. And there's only a small amount of these regions that we need to be concerned about when we're looking at for breeding material. We can do GWAS on survivability and identify some really strong candidates of genes that we can go back after with targeted modifications and understand what these genes are doing. And so by combining these PacBio high quality references with large-scale Illumina resequencing, it really leads to unique biological insights. You can't just do anything with a genome alone anymore. I just want to remind people of that. A genome is a genome. It's a representation of genes and sequence. You have to be able to do something with it from the science side. So I want to talk a little bit about cotton. Cotton projects are managed here by Avinash, and then with a lot of effort from Jerry's group and James' group for sequence and analysis, production of genomes. A long time ago in 2012, we produced the first diploid cotton genome, which is called the cotton D genome. It's right here, looks like this, doesn't produce a lot of fibers. And historically what happened is about 1.5 million years ago, A and D came together to form this thing that we think of as cotton in the fields, which has a lot of fiber, and that's what's planted as a crop today called the AD genome. It's a tetraploid. It's mostly inbred tetraploid. So we started working on a many tetraploid cotton genomes project where we identified the five remaining known tetraploid cotton genomes and sequenced them all. This with this cast of characters here. And so this is just what some of the genomes look like. These are all CLR genomes. I'll come back to HiFi in a second here. So they got better over time. This is early on RS data through SQL data, through high coverage SQL and into SQL 2 data years. And this is just what the cotton balls look like here. GH and GB have been selected for big fiber. And so in comparison to some of the other ones, like this is the published cotton genomes, like what were we thinking? 444,000 contigs. How are you going to do biology with that? Anyway, things move on. One of the things we noticed when we were working on this project was that it was hard to tell them apart. They looked pretty good. This is collinearity again, just on some dot plots to show you that for the most part, they look pretty similar considering they're over 630,000 times 2 years diverged. This is a nice plot from GeneSpace, which is our pan-genome program we're working on right now to derive plant pan-genomes led by John Lovell at HudsonAlpha. And you can see genes are pretty well conserved all the way across the A subgenome. These are the diploids, including diploid D, and then out into the D subgenomes. Really nice conservation. Max SNP rate's about 1 in 160 base pairs. You might know that as pretty close to the maximum variation that's in the human population. I mean, incredibly inbred for a wild plant population. So there's some minimal gene differences. And so with little variation, how do we actually improve cotton as a crop? And so we started a project that's now we're calling the many elite line cotton project. No, we don't have a real name for it. So we are sequencing elite line cotton coming out of breeding programs across the US and the world to identify changes that are linked to these advanced agronomic traits we have in the field. This is just an example of an Australian line that has this cochra leaf trait, looks like okra, versus cotton that is useful in regard to photosynthesis. And so we sequenced several elite line genomes and getting pretty good PacBio genomes. We got this down. We're good at this. 300 contigs, looking really nice. Lots of continuity. This is our most recent reference version of cotton with 40 meg contiguity coming out of early long read SQL 2 data. And it looks really nice gene-wise, getting complete gene annotations from there. So we've been looking at SNPs and indels in these elite lines versus TM1, and they vary about 500,000 SNPs or so, and indel events about 1 per 500 kb. Overall, there's almost no differences between these. This is just a Circos plot to show that some of the highlighted areas, we've got a few differences in SNPs, and a couple of chromosomes look like they've got some interesting variation in indels and SNPs. But for the most part, we're really looking for needles in the haystack. And can you imagine doing this in 444,000 contigs? So my take home number 8 is we really need the resolution of accurate whole genomes to be able to identify these agronomically relevant traits in bottleneck crop species. Is cotton the exception or the rule for major crops? That's a take home assignment for you to figure out. So brief history of de novo genome sequencing in plants. Same plot you saw before here, although I've added my smiley faces so you can see how sad I was from 2007 to 2014 as we worked on genomes. And this is approximately the cost of sorghum genome over time. And so with SQL 2, with HiFi, we're down to less than $10,000 a genome, and I've given it the coveted 2 smiley faces award. We're very happy these days as we work on these. So I'll tell you a little bit about sorghum and some of that work. So this is grain sorghum, produces grain. People eat it. We also feed it to animals, and it's planted around the world as a food source, particularly in developing nations. This is, this is biomass sorghum, and that's super tall. and it's in a perpetual juvenile state, and we use it to grow as a biomass source. And so we consider this a transitional biofuel. It's planted like an annual, uses existing agronomic infrastructure, and we can make lots of biomass with it and convert it into things like liquid fuels. So there's lots of people who contributed to this. Rachel's led a lot of this, and Jerry and Chris in the assembly group, and Shu on the annotation side. Sujan, Adam, and John have done a lot of analysis for these projects. And of course, much of this data has been produced by Jane's group. So this is what sorghum genomes look like over time. This was our finished Sanger 2000, you saw the paper, 2007 there, 0.2 megabase continuity with 13,000 contigs. This is a sad, sad version of what an Illumina sorghum genome looks like, 50,000 contigs. It's sad and depressing. And then we did a lot of work to finish, used Concept among other things, and brought this thing up to 1.3 megabase continuity. This is the first RS2 versions of sorghum, and at 4,000 contigs and a 400 kb contig N50. We were so happy to see this at that point in time in 2016, and then moving up to 17.7 on the sequel. This is what HiFi looks like. So this is a recent version of our reference. Again, same genome, 89 contigs, 50 megs, integrated with HiC, looking like a really nice genome. So HiFi, major genome advantage for plants is increased ability to assemble difficult regions and repetitive regions, detangle haplotypes and outbred and polyploids, which I'll tell you a little bit about in a second, and then also greatly decreased computation. This cannot in any way be emphasized enough. This allows us to run multiple versions, multiple assemblies, evaluate the results, and really optimize it for an individual organism and produce high quality, accurate consensus sequence and accurate assemblies out the other side. Just to show you synthetic blocks of genes, looks pretty good. We've made some improvements even over our improved Sanger for 3 version. Looks really nice. We also added 6,000 genes since version 1 and another 2,000 private genes. We're starting to show a lot of private genes now because we're adding extreme amounts of isoseq data and new annotation improvements to really get at that. So this is just one HiFi genome. Think about what can we actually do with this stuff. So we've been working with collaborators, including Todd, Jeff, and John, to really build the sorghum genomic infrastructure based around high quality pan-genomes and lots of resequencing data. This is just an MDS plot here showing you genetic relationships. Every one of these little circles is a resequenced sorghum genome. Every one of these larger squares, circles, and triangles, these are reference genomes that we've sequenced now for sorghum with long read and HiFi to try to build this large scale reference genome. And this combines a lot of different funding sources and efforts. And it's really the only way to build this kind of infrastructure these days. And so just to give you a pan-genome sorghum example with 14 different variants here, our references at the top, these little guys here are private genes that are private to 2 separate ones of these accessions. You can see in our reference, we actually are missing 4 genes out of 17 genes that are contained in this interval with these, looking at this 14 pan-genome. You can see kind of spreading it out over there. What it really looks like is, I'm not really sure this was the best reference to choose. It's a reference that's used a lot because there's quite a few other sorghums here that have 15 of those 17 genes present in them. And then you get into the subpopulation down here and you're really in the 11, 12 range in there. And so this is the reason that we're pushing towards pan-genomes for plants, because plants have lots of gene level variation. Even this is 13 genes, a tiny, tiny region we're looking at here. Obviously in humans, you would not want to have these genes missing. And humans are much more constrained and contained than plants are. So to take that into a pan-genome example, this is a great, really useful agronomic gene, LGS1, which in sorghum is a deletion which gets rid of strigolactones, which are these kind of sugary molecules that get released by sorghum to encourage growth around its roots of bacteria. And it so happens that a clever little plant called striga has co-evolved with sorghum over time as a parasite, and that grows with the sorghum and eats these strigolactones. And so an insertion deletion to break this up actually reduces the striga growth on sorghum. And you can see of our little 14, I made this a 15 pan-genome reference version here that we're missing in PI6, that gene there. And so this is a reference deletion, and that's why I can show you this data, because we really haven't gotten to the point of being able to analyze everything for this. So you can extend this into other resources. We have our present-absent variants that we built with Illumina-based sorghum pan-genomes, and you can see in subpopulation world that 4 here has a reduction in this this gene, this LGS1 gene. And then if we go back to look at, say, 12 to 1300 resequence libraries, looking at just the gene, you can see all coverage variants across this region. And here, this is the insertion deletions that that are described in the paper. There's also another one up here that really also can contribute to knocking that out. And this one's across a whole set of different subpopulations. And so what this whole genome, high quality genome combined with low quality resequencing and our medium quality pan-genomes allows us to do is look at all the genes that are present in the subpopulation and do this kind of analysis and really identify genes that are useful in an economic sense. And we're not limited anymore to the single version of that pan-genome. So these high quality pan-genomes provide access to the most of the entire diversity of the species. So to give you another example of a plant, you might be thinking it's these bushes and trees here, but it's not these bushes and trees here. This plant from a recent project that we're working on actually lives here out in the ocean. And this is a seagrass in one of these coastal interface zones. Seagrasses are pretty amazing, and they are distributed around the entire world. And you can see by this nice plot here around all the coastlines around the entire world. And they do a lot of really exciting things for us. We sequenced the first Zostera marina quite a few years ago. It was published in Nature in 2016, led by Janine Olson. The important part about these is they're significant blue water organic carbon sink. They also do a lot of nearshore ecosystem services and promote species biodiversity. They're one of these threatened areas by changing temperatures in the ocean, changing weather patterns. And they really do an enormous amount for the planet. We know almost nothing about how these things work in the environments. And so this is a project to try to understand the diversity of these plants and how these plants interact with the microbiomes in these regions to do carbon sequestration. One thing that's really interesting about these seagrasses is they're actually monocots from the land that have returned to the sea. And they've done it over and over and over again. And so we can build up some information from the biology of that. So 6 years of sequencing progress. The old stuff published in that paper looks like this. This is our new PacBio. This is based on CLR, last year, HiC plus haplotype correction. And it's a really tiny, cute genome of 250 megabases, so we can do a lot with it. We've currently started working on another project to expand this out and look at other species here. And this is Cymodicea. It's another cute little one that we feel pretty confident about. We also have bigger ones like 3 gig. These are outbred genomes. They're difficult to access, and they're really working in environmental contexts. These aren't cultivated at all. And then Thalassia, 4 gigabase genome. And I'm going to show you results from that here on HiFi. So this is the next seagrass genome, Thalassia. It's a 4 gigabase outbred monster. So we sequenced about 60x of HiFi coverage. Remember, about 30x of haplotype here. Assembled it with HiFi ASM. We got a 4.9 gigabase primary and a 3.8 gigabase secondary with a 483 megabase contig N50. We were real excited about this, not just because this project is unbelievably complicated to do with CLR and in our previous methods. We would spend a year sorting this thing out and making it into a genome people would want to use for something. We had 5 misjoins, and so it turned out with the HiC it looks really nice and sweet. We ended up with 9 chromosomes and 24 contigs for this genome with a 371.6 contig N50. I'm not sure that's the greatest contig N50 everyone's got in the world, but I'm pretty sure that that means that we should have 371 PacBio buttons for the Megabase Club, just for one chromosome here, or one genome here. Anyway, this is what it looks like relative to sorghum. You can see how cute and small our orange sorghum genomes are, chromosomes are relative to seagrass. And it's very exciting to be able to do this kind of work. We ran into a BWA-MEM issue, of course, which turns out to be rarely known, but documented that BWA-MEM does not work with contig greater than 536 megabases. So my take-home number 10 here is HiFi enables what once were impossible sequencing targets. I mean, this is a wild, outbred plant and a complicated one, and it's amazing to see this kind of work. So what about our other species that we talked about? HiFi for cotton. Here's a HiFi for cotton slide to show you that we're continuing to produce more reference genomes for cotton elite lines at 90 contigs with nearly a 70 megabase contig N50. Looks pretty sweet. If you compare this back against, this is 2019 and 2015, these genomes were published. Start to compare this back against this, what you see is that the old ways of doing sequencing for reference genomes, you should never do them again, ever. Okay, this is a 2019 paper showing 10,000 fragmented genes here versus these higher quality CLR, and then our higher quality HiFi genomes there. And so you can imagine what life was like working when you're trying to do anything with microbiology and cottons back in 2015, 2016, 2017, really difficult. How about Switchgrass? So we started something, Switchgrass pan-genome project. Why not? You know, we've got all this data, huge amount you saw I presented earlier that we can use with these new high quality genomes. So that's a 70x HiFi genome, 1824 KB library, which is pretty much our standard these days, assembled HiFi ASM, a 1.3 gigabase primary with a 58 megabase continuity. This is looking really quite sweet. You can see on the alts, we don't quite assemble them as much and have as long a continuity because we are still dealing with a complex outbred repetitive grass here. Just to show you what this looks like on some of our V5, which I showed you earlier, 2 contigs for chromosome 1K and chromosome 1N that look really quite decent. You can see differences. Obviously, we're improving some order and orientation in paracentromeric regions quite a bit here in the centromere for N, which has expanded centromeres of getting data, and a little bit of stuff that we've got to really look at up here on the P arm. But you can see that, you know, I have real hope that we're going to be able to do this thing that I'm now calling the pretty promising pangenome of panicums. And I encourage you to say that several times fast. And so, yes, number 11 here is even for Switchgrass, HiFi makes pangenomes possible. It's unbelievable coming from somebody who spent 14 years now working on one genome sequence for Switchgrass. So a lot of our plant projects are available here at Phytosome, phytosomenext.jgi.doi.gov. We've got 236 plant genomes available there. We've got lots of tools for comparative genomics. And this is, I encourage you to take a look, and that's where a lot of our data releases come out. If you're interested in working with the JGI on JGI DOE relevant projects, please visit the jgi.doi.gov user program. So we work in lots of areas, not just plant genomes, and you can put in a proposal and work with us on these kinds of projects. And so we have the technology, and the question is, you know, so what can we do with it? Well, one thing that we could do is a genome for every child. I know it's hard because we're still on exomes for every child, but really it's not a great leap from WGS to HiFi genomes for every child. And I think based on discussions that are happening yesterday with Evan, that once we have these hundreds of reference genomes that are high quality, haplotype resolved, it's going to really bring down the cost of being able to do this on an individual project, or for an individual person to be able to generate a genome reference we can use to identify all these variations. Now, me personally, I don't want to see what my genome looks like because again, who knows what it is, and I certainly don't want to see what I gave to my 3 girls, that's for sure. Notice they were all girls, no Y. Yeah, anyway, end of the line for the Ys. So what we can do in natural environments is really understand individual species level diversity. I showed you all this stuff about Switchgrass. Switchgrass is just one plant in the prairie ecosystem. This is a really nice, pretty picture of a prairie. You can tell it's artificial because a restored prairie because it looks so nice. Because when we restore these, we restore it like with these monoclonal collections of really pretty things that we like to walk through. But Switchgrass over time has been decimated. This is Illinois again. We're down to 2 little regions of Switchgrass left in really natural prairie in Illinois. And so that's what's going on with these ecosystems over time. And these are really critical for decisions on managing what remains of these Earth's natural systems, being able to understand the biodiversity. We can investigate important ecological systems that are impacted by climate change. I gave you an example of seagrass and the seagrass ecosystems. There's plenty that you can think of, including obviously coral reefs. Here, where they're really important for climate biology interfaces, where we are trying to understand how climate works, but we don't even understand how diversity works and how these ecosystems work on the level of collections of populations. This is arboreal forest here. Projects working with Dave Weston on sphagnum. DOE has a really interesting project where they're putting ecochambers out to be able to increase CO2 and temperature over time, and then be able to understand what happens in these commensal relationships between microbes and plants and trees and the environment. We can understand managed diversity and forest health. We've heard a lot about forests this year, a lot about how do we do restoration, what's going on with the forest under drought and climate change, and we don't know nearly anything about what the diversity of most of these species look like and how that diversity changes over with climate impact. And so we can sequence genomes for these now. We can look at diversity. It's just not that big of a deal. The genomics part of these projects are now small relative to the effort needed to be able to even track these things over time. And then a personal research effort of mine is to think about remaking agriculture to be really sustainable and lower impact on the planet. This here is a nice picture of corn. Corn and lots of our modern crops have been bred over time for one thing: it's yield. It's sort of humorous that it also happens to be the same thing that suggests that we slow down, but in this case it means maximizing yield at the cost of anything else. And in inputs, for example, nitrogen runoff, and then and emissions from nitrogen farming. We really have to think about how do we fix this stuff going forward and remake our agriculture. And this stuff really makes me sad. That's a big blue sad smiley face for this effort. So one of the kinds of things we can do in integrating other kinds of data in there, this is projects working on with UNL, integrating high throughput phenotyping. This is low N and high N nitrogen sorghum phenotyping. We've done extensive transcriptomics across growing season here and identified genes that are really important in important timeframes for these, where the plants are uptaking nitrogen, using it, and then using it to produce the end product. We can go in and we can, with targeted modification, we can mess around with these genes and we can evaluate what's really going on in the plant to identify new targets. And this is really whole life cycle analysis of nitrogen responsive genes in plants, way more detailed than anything that's been done to date for these kinds of projects. And so we can bring these things together to really understand the biology of crops and breed for earth system impact and resilience. Instead of just breeding for yield, we can take the whole system into account. We can also develop and use perennials and cover crops for things like, in our case, bioenergy, or for food that minimize impact inputs and reduce nitrogen and nitrous oxide leakage. They're big problems and they require large-scale experiments and cooperation across many areas of science and industry coming together into new priorities. So I encourage you, we've got the tools and technology, and we should get together and do something about it. To finish up here, there's some email addresses at the top. If you want to email people, I wanted to mention that a lot of our biomolecule work is done by Arizona Genomics Institute and Rodwing and Dave Kudra. Dave's email is on here. This is our group at Hudson Alpha, the JGI group at LBNL, and Greg Cooper's group contributed a lot of the human work. Really pleased and happy that a lot of funding agencies have been able to fund this work and have trusted us to be able to do things with taxpayer dollars or contributed dollars to really make impact with genomics on the planet. It's been a really exciting ride the last 5 or 6 years with PacBio technology. So thanks. With that, I'm going to stop sharing my slides and go back to Jonas for questions. Hey, how'd I do on time? Hey. I'm sorry, I'm not getting the audio back from Jonas. So let's ask some questions. So so where does coverage level off? That was a question here.
Okay, sorry about that. I lost my window, but I understand you can hear me. So I apologize. What a great talk. And thank you so much. Yeah, we have quite a few questions coming in. So you talked about one of the take-home messages was more coverage gives you better assembly, but at some point that's got to level off, I would think. Did you do a take?
Yeah, I did. Yes, I didn't do one of those really nice, like we've taken these to 70x and I've shown you all the assemblies that come out of there. But yeah, for all of our plant genomes, and I suggested also for, really high quality human genomes, really we're targeting 30x a haplotype. And so it's super important if you're doing haplotype specific resolution to have something like 25 to 30x. You can get a decent assembly with HiFi out of 20x, especially if you're, interested in kind of getting big contigs out. For sure. But when we're putting together reference genomes, we find that by pushing it to something like 30x, we're really getting these big, you saw 2 contig whole chromosomes coming out, and really have to be high enough to account for all the variation in the sort of in the length in the library and distribution in the libraries and all that kind of stuff. You really have to push it up a little bit higher than that.
Yeah, and I mean 371 megabase contig N50. I've already contacted my colleagues. We'll definitely make a special button.
Special button?
1 megabase.
Okay. It's got to be a real big one though, not like one little like 1 megabase, 10 megabase called button.
That's amazing. So related to that, so you had the cost numbers for the human genome, and so was the sequencing cost, and then there was the assembly, sort of mid-level assembly cost. And did that include the still the HiFi calculation? In other words, would the Sequel 2E now putting the HiFi data out directly bring down that cost fairly significantly?
Yeah, I think probably for some people, definitely. I think most of that cost is in machine time and machine cost and service contracts. And you got to remember that we're producing 4 chips a week per instrument, and so anything that can make that increase that number of chips for producing the same amount of HiFi out the other side, that's going to really knock that cost down quite significantly. As opposed to, just saying that, there's sequencing cost, the HiFi calculation cost for us is not really particularly a big burden anymore relative to what it was in the original HiFi. As HiFi started to get introduced, I mean, these days, it's not super expensive computers doing these calculations anymore with the improvements that have happened in the algorithms for the HiFi calculations.
Yeah. Thank you. A few more questions coming in. Do you use transcriptomics and isoseq to gain complementary insights in genome annotation?
Yes. Oh, for sure. Yeah. So most of our genomes these days, we put something like 12 to 20 million full-length cDNA from isoseq protocols on to help, and that really I didn't have slided. Sorry. Really increases our quality of annotation and alternative splice annotation. For specific for plant genomes because of the complexities of alternative splicing, for sure. And then I didn't show a lot of transcriptomic data, but for many of these projects, we're really generating significant amounts of targeted transcriptomics, which we can use to tease out some of the biology of how those genes actually work in the organisms.
Mm-hmm. For diploid, about 500 megabase, let's say plant genome, which assembly methods do you recommend? And then also HiFi, BioNano, and HiC for chromosomal assembly, or is HiFi 25x only? Is that enough?
Sure. So for diploid, smaller genomes, yeah, I'm going to go with HiFi ASM right now, assuming the HiFi data is good, for sure, because the advantage of that is really getting at kind of trying to walk the path in 2 separate ways and split out individual haplotypes. And I did want to say, also thanks to people who are working on tool development for this, because the tools have gotten amazing and they get even better every single month. So keep that in mind when you're working with HiFi data right now. It's like literally changing the wings on an airplane as you're flying, and hopefully you stay up in the air. In regards to, definitely diploid, in regards to the question of what our methodology is, for most of our genomes, we're really HiFi 25 to 30 x a haplotype plus HiC to be able to do validation and orientation. For really complex genomes, we're generating mapping resources just like we used to generate in the old days. But for something that's like a 500 meg genome where you can expect to get half a chromosome out at a time or 100 contigs or less, then yeah, HiC is perfectly fine. We would typically not use BioNano in those kind of instances.
Mm-hmm. And do you polish the HiFi assemblies with Illumina short reads? The question is many.
So yeah, that's a great question. So the 30x coverage on HiFi really gets you down to a small number of indels relative to what the old CLR approaches did, and polishing with Pylon and cleaning them up. However, for us, typically we almost always have Illumina data and we do almost always use it. But we're producing these reference genomes that we hope will hold up over the next 5 to 10 years. And we're going after the 500 genes that might have an indel in them. And so you've got to think about it that way. What's your gene rate versus your genome rate? And that's really, you got to make that kind of question, that kind of decision on a project by project basis. Think about how much effort you want to go to. I always encourage people, if you have the data, to do it, because in annotation, it's really easy to break genes if you have an indel in it. And keep in mind, the indels are biased against the places where genes are and all that stuff too. So yeah.
And then you polish in a sort of filtered and sort of very specific way where it maps, uniquely into both
Yep.
applications.
Yeah, we've always, right, for Illumina, we've always only polished uniquely, mapping positions for sure. And so think about that. The advantage in the HiFi world now, the reason that we've switched wholesale over to HiFi, other than all the other things that we already talked about today or yesterday, is the fact that not only do we know that consensus is much more accurate in the regions we can map Illumina to, we also know it's much more accurate in the rest of the genome than that we don't normally necessarily map Illumina to and polish and fix. So that's a real advantage for us working in complex, repetitive organisms. We know our repeats are also more accurate.
Yeah. The next question, so you showed that the references are really good for, resequencing and seeing the, in space and in time, the changes and so forth. And is that mostly restricted to SNPs and indels, because the large scale population sequencing, that's done with Illumina, and so you might not be able to be sensitive to structure variants?
Yeah, definitely. I mean, I think we've called structural variants and have called structural variants and shown, population and genomes of structural variants for different plants. For example, Soteria, there was a paper that came out a few weeks ago. And you can definitely do some structural variant work with the Illumina. It's really restricted in plants to the to kind of small, mostly inbred organisms, where we can build bigger pieces and then use those bigger pieces to call structural variants. There is, people have done plant work, and you can definitely call structural variants in Illumina with that kind of data. But where we think we're getting the most power from is building many high quality references, identifying all the variation that we can find in the high quality references, and then projecting that into the resequencing data, rather than the other way around, where you're just throwing resequencing data on trying to figure out if there's a structural variant or not there. And so we think of it as kind of the top down and then the bottom up approach, where we increase the accuracy of the annotation, the accuracy of the analysis for the resequencing data by aligning it against the local reference, which bumps up our accuracy there and impact, and then projecting those structural variants from the high quality pan-genomes into that resequencing data.
What do you think about doing a population genome sequencing experiment with HiFi, but doing that with low coverage per individual, right? So to keep the cost down at the moment, and say 3 to 5x coverage per sample, and then so analogous to the 1,000 Genomes project initially, like it was done, right? It was like 7x coverage per individual, but you had 1,000, so you had 7,000 coverage over the population.
Yeah, so I think that's an interesting approach in the plant world. I think the problem that we have right now is, if you're not going to pool them together and you're going to make them as individual libraries, then it's a balance, because you've already you spent a lot of effort to get to the point of getting high molecular weight DNA that's suitable for HiFi, making a high quality HiFi genome that you can get 3 to 5x coverage is distributed out, and then you're going to tell me that we shouldn't run one more chip on that to be able to produce a haplotype resolved whole genome that we can actually identify all the variants from using existing tools? I don't know. It's a trade-off. And so I think there's options. But for us, once we have a haplotype resolved genome, we can annotate it. Mm-hmm. So we can really annotate impacts and bring in the issues of these genes that aren't in the existing references and bring those in, as opposed to just structural variant identification. I think it's great if you just, you want to do large scale structural variants. That's a great methodology.
Yeah, and certainly I would hope that the future is going to be there, right? As the costs come down further, then you'll just do that on larger and larger number of samples, right? So, right. Great. Well, thank you so much. That was just such an amazing ride of all the different things and really amazing progress. And lastly, I can attest. I've switched over to sorghum-based popcorn a few years ago, and I find it, personally, I find it a lot more tasty than the conventional one. So thank you so much, Jeremy. And what a great talk. And thank you for speaking at our.
Okay, I appreciate you giving me the opportunity. And thanks for pushing on sorghum. You know, sorghum is the old maize. We used to grow it across the U.S. Before corn. So encourage you to pick some up popcorn. Thanks, Joe.
Thanks so much.